can't be here right now. And welcome back. Yay. Hopefully you guys used your time last night to at least contemplate what chapter 26 is about. Hopefully you got to the structure of a nephron so that when I draw one for you right now, actually what I'll do is I'll draw a stick figure. Cool. Right, stick figure, put some kidneys in here for you. Kidney. Travel down to a blood. Whoops, that wasn't supposed to happen. We'll figure that out. Bladder there, yay. Cool. Generally speaking, we're trying to figure out what we're going to be doing with these two structures here. And what I was hoping people would do is they would get to a place in their reading where they saw something. Yeah. Wow. Okay. Surround this now. This. Yeah. We'll see in a couple minutes why this is actually so important. Whoops, that shouldn't happen. This happens right here, you die. So you probably never want that to actually happen. Okay, so just bad art you can learn by. But the idea, of course, is every single moment of your life, all of the fluids inside of your body, not all at once, but over the course of some period of time, actually have to pass through these structures here. And ultimately, what I need to be paying attention to is how Mother Nature is going to regulate the flow of oxygenated blood. This is the first time we've ever seen something like this. Oxygenated blood sending materials inside of this space so that we can begin to filter it. That's where I'll just leave the introduction for right now. So send fluid into this space here where ultimately, at some point in time, your body needs to make the decision of what should stay and what should go. And that's really the goal, right? If last night you believed what I said, that your lungs take care of pretty much carbon dioxide, your kidneys are taking care of essentially every other electrolyte in your body. So to state their significance, well, let's just say they're important. Take a look here. Regulation of blood ionic composition, regulation of blood pH, regulation of blood volume, regulation of blood pressure, regulation or maintenance of blood osmolarity. You can read this stuff yourself, okay? But production of hormones, calcitriol, as well as erythropoietin, regulation of blood glucose levels, so on and so forth. The list goes, it's longer than this, but these are the major responsibilities of your kidneys. So let's just say losing both of them means you don't have a life. Now, the external anatomy is pretty straightforward. In fact, this is a 30-second dissection inside of a lab where you just take a look at a kidney and you cut it into a frontal section. But what you would see is there's an indentation where the ureters emerge with blood vessels and lymphatic vessels. Mother Nature creates this space so that she can create pressure just deep to this renal capsule, which is a continuous outer coat that protects the kidney itself from outer trauma, but it also maintains the pressure inside that space. It has a renal fascia. It's a superficial layer, thin connective tissue, which anchors the kidney to the back of the wall. You are in the retroperitoneal cavity if you are looking at a kidney, okay? I should say if you're actually palpating a kidney. So here it is, a frontal view here. Here's those two really bad cartoons are. That cartoon I drew a moment ago. Here are your ureters descending down to fill the bladder. And here's what we'll be looking at inside lab. Okay, this is a really old, really dead kidney, where the adrenal gland or suprarenal glands are superior and medial on both kidneys. Notice the left kidney is slightly higher. Okay, but what's going to happen is we need to take this apart and begin to look at what's going on inside. And here's what's going on inside. So if you've gotten through chapter 26, you realize this is actually at the end. This is figure 26.20. And what I want you to see is that inside of this kidney, what you do is you decorate about 1 million nephrons inside of this space, and their responsibility is to filter that blood moment to moment. Okay, you didn't see what was actually happening here, but each portion of a nephron, okay, is going to determine which molecule is either reabsorbed or secreted. And we have to pause here. Reabsorbed means it will be passed out of the tubule and be collected back into your circulatory system. Secreted means it stays inside the tubule, and ultimately, if it's not collected back out at some other point in time, it is converted into urine and exits the body through the urethra. So for whatever reason, these words seem to give pause to students. So I just want to make sure. So reabsorption would be, let me draw something for you here. Reabsorption would actually, the arrow by the actual mechanisms of transport will move materials outside here. So it's missing inside our cartoon will be blood vessels. If it stays inside the tubule, it means ultimately this will be secreted and this ultimately becomes urine. So what are the parts? Every single nephron has a proximal convoluted tubule. Yay, convolutions. We'll draw this again for you in a little bit. We'll have a loop of Henle, which has a descending limb and an ascending limb. And notice that the limbs are different. The thickness of the ascending limb increases. Then we have our distal convoluted tubule. And then we have our collecting ducts. And we'll see how those all work together. So let's think about the cortex, where we're going to find those nephrons. Is that a question, Janelle? No? Okay. So the renal cortex is the most superficial area, and the renal medulla is going to be these cone-shaped structures we're going to call pyramids. When you actually take a look at what that looks like, here we've taken a frontal dissection away. So if here's the capsule on the outside. Going deep to that, this is the cortex. And here are those pyramids I tried unsuccessfully to draw for you last night. And the reason why they cut the kidney this way is you're going to see what they do is each of these pyramids literally creates a cone-like shape. And that cone-like shape in 3 dimensions collects materials and transports them into these areas here, which are called calyces. So you see this yellow structure here? That yellow structure is that nephron. Now imagine you have 1,000,000 of them inside of this space. And each of them are draining materials that will ultimately go from the minor calyx to the major calyx to the renal pelvis to ultimately exit through the ureter. You're like, that's no problem. I got this. This is so easy. And it is. Okay? For at least this piece of it. Okay? Mother Nature's figured out a way to filter materials. You don't look so convinced, Elizabeth. You're like, no, there's going to be something different going on here. Of course there is. Figuring out how to regulate the movement of fluids in the body is actually really difficult, right? So, and by that I mean, you're going to see there's a series of mechanisms we have to sort of develop here. Okay.
That's where I get confused.
That's okay, but that's okay. Hopefully I can help make it less confusing. Okay, so what's going to happen is this. We have to send blood to each one of those nephrons. Okay, and each one of those nephrons is inside the cortex, which means, although the kidneys constitute less than 0.5% of your total mass, they receive between 20 and 25% of your resting cardiac output. This value drops as you increase your physical activity. Okay? Sympathetic responses actually decrease this value because the blood is needed someplace else at that point in time. And that's why it's considered resting cardiac output. You're going to see that there's going to be a left and a right artery that'll feed the kidneys, and we're going to branch into specific destinations. Segmental, interlobar, arcuate, interlobular, okay? And each is going to have one afferent arteriole traveling to each nephron. Okay, so I want you to think about that for a second. Remember yesterday I just said that your kidneys are just one big fancy capillary bed? This is what I meant. Remember back inside the chapter 21, we said a capillary bed has an arteriole. Well, our nephron will have one arteriole traveling to it. Okay, that one arteriole is going to be where we actually diffuse materials out of the plasma of our blood and into the capsule. And here's the tricky part. The other side to that capillary bed is still an arteriole. It's an efferent arteriole, and we'll see how that works in a second. Then the blood will, of course, pass out, and this is actually how we will collect the materials that need to be reabsorbed. So it seems like it's an inefficient system. Push materials into these tubular structures, maybe a subset of it is reabsorbed, maybe a subset of it is secreted, but ultimately it has to get back into your bloodstream. And it does have to get back into your bloodstream, because this is actually how we balance the ionic concentrations of material inside of your body. Now, a moment ago, I told you that the arresting cardiac output is what determines the filtration inside of a kidney. The renal nerves are part of the sympathetic autonomic nervous system. So now when we need to decrease filtration rate, you're going to see that the vasomotor nerves are what regulate that blood flow there. So sort of similar to what we saw at the end of chapter 21, the vasomotor nerves traveling out to the arterioles will vasoconstrict, okay, or relax, depending upon what's going to happen there. But the sympathetic nervous system does not cause dilation. It causes vasoconstriction. Let's see how that works. First things first. Here's your kidney. Here's your kidney dissected, okay? Kidney dissected. Let's take this small piece of the kidney here, and let's blow that up for you here. And here's what I've been trying to draw for you. Look, here's our afferent arteriole and our little red lines. Well, the red line goes in and a red line comes out. This is what I meant when I said an afferent arteriole travels in and efferent arteriole travels out. What color does that efferent arteriole become?
Purple.
Fantastic. Thank you for somebody answering. I was going to sit here all night long till somebody said something, right? Yeah. The point is that even though this looks like it's a portal system, it's not truly a portal system the way we've appreciated it in the past. This network that's being created here, this network is what's allowing us to reabsorb materials back into the circulatory system if it's been pushed out of the actual tubular structures themselves. Remember, anything that's inside the tubule stays inside the tubule until it's moved out, okay? And if it doesn't get moved out, it ultimately will be secreted out of the body. Sort of like Vegas. It happens there, stays there. At least that's what people say. I've never been. So, now the idea, of course, is this, is if you have 1 million of these, that means you actually have 1 million peritubular capillary beds. It means on some of your nephrons, you'll have 1 million vasa rectas. So when we start thinking about regulation, this is what we're actually doing when we're thinking about how blood will flow into this space and ultimately how you recollect it. And the way to think about that is on the far right side of this slide. Starting at the top, okay? You'll follow the renal artery into the segmental artery. Segmental artery travels into the interlobar, arcuate to interlobular, which then becomes that afferent arteriole. And that's right here. Becomes a glomerular capillary bed, and materials can be collected here again and then sent back out through the efferent arteriole. We then travel through the peritubular capillary structures where materials are reabsorbed along with and then transported down to the interlobular, arcuate, interlobar, and renal veins. So yeah, how many times have I actually told you a specific circulatory route inside AMP2? Only for the heart, right? Notice we just went through all the steps here. What do we think this means?
I have to know it.
There you go. Look at that. You guys are gonna, you guys are going to save lives. I'm starting to get hope here. Cool. So, and unfortunately, the only way to practice it is to sort of draw a quick cartoon like this, sort of notice where they're traveling. Each is a specific place inside the kidney, and you'll see that inside lab or on presentations I put together for you in the future. So then, let's take apart the functional unit. There's the renal corpuscle, which is actually where you filter blood plasma, and it's made up of a glomerulus. And we'll take a look at what's actually happening inside here. Whoops. There are two types of nephrons inside of your body. You're like, damn it, I'm only wrapping my brain around the fact that there's a nephron, and now you're telling me that there's two types of nephrons. The vast majority of nephrons inside of your body are cortical nephrons. They're found inside the cortex of the kidney. The remaining smaller number, but maybe more important, are the juxtamedullary nephrons. We use the juxtamedullary nephrons when we are dehydrated or when you have too much water inside your body. The moment-to-moment maintenance of what is going on inside of your body is regulated through the cortical nephrons. Okay? Now that might seem like a big deal, but if 90 to 95% of your nephrons are cortical nephrons, it means it's sort of, if you think about the consequence of what you see here is the smaller loop of Henle that you see here means that there's less surface area, which means there's less opportunity to actually reabsorb materials from the actual tubular structures themselves. So we're going to use them at distinctly different times, okay? But Mother Nature is going to have to figure out when. You already know when she's using them, okay? Way back when, inside of chapter 18, we were releasing a molecule, and that molecule is ADH, right? And as you increase the concentration of antidiuretic hormone inside of your body, you switch between a large number of cortical nephrons to a larger number of juxtamedullary nephrons. And in doing so, you increase the likelihood of trapping sodium ions in the body so that water will stay inside the body. So that's a really good way to think about this. If you have very dilute systems, you don't need to regulate your body through ADH as much. But as you become more and more dehydrated, you need to regulate the total amount of water inside of your body, and therefore you increase the number of these juxtamedullary nephrons. Again, the major distinction is they're closer to the medulla. The other way to tell during a test type question would be that if you look down here, this entire structure is the peritubular capillary bed on the cortical nephron. Over here, anytime you see somebody referring to a vasa recta, the vasa recta means you are talking about a juxtamedullary nephron. Okay? So that is your tell, okay, when you're discussed, because everything else is roughly the same except the surface area. Okay? You'll see what that actually does to concentrating the materials that are inside the fluid of the filtrate. So what is that glomerular capsule then? The glomerular capsule is made up of two layers. Look, repeating pattern, must be at least number 10 for this summer, right? We have a visceral layer and we have a parietal layer. We have a visceral layer, which is made up of a series of cells, which are called podocytes, and they wrap around a single layer of epithelial cells that make up the glomerular capillary bed. So if you wrap your hand around in a circle like this and you put your fingers in a distended way inside your hand, your fingers would be the endothelial cells and podocytes surround those. And that makes up the visceral layer. The parietal layer is made up of a series of cells that just create the wall of the capsule. Fluid's going to move into this space and it's going to push out. And that's actually what you see here. In red, oxygenated blood is traveling in through that afferent arteriole, traveling into the glomerular capsule here. And inside of here, you actually have those endothelial cells that are covered not only with podocytes, but also these cells here, mesangial cells. And the mesangial cells, notice there's another population of mesangial cells here. These mesangial cells are detecting the stretching effect, how much pressure is inside the capsule. Mesangial cells are responsible for producing nitric oxide. One more time for effect. Mesangial cells are responsible for producing nitric oxide. And together with this piece here at the back end, I guess it's more the anterior end of the capsule, right? This is going to be the macula densa. And there's communication that happens as a result of the pressure inside of this capsule and the concentration of ions that can be detected at this thing that's called the macula densa.
So in that area, would you have just vasodilation or because of the nitric?
No, great question. Notice what's actually happening here is that afferent arteriole. Do you see these yellow structures here? That's the vasomotor synapsing on the smooth muscle of the arteriole. And what happens is the regulation of the total amount of pressure in here is how fast is the blood coming in, and then how much dilation has to happen, or how little dilation actually has to happen inside the capsule. Okay?
Okay.
Cool. Because that ultimately, you know what we're going to do here. We're going to create a pressure gradient, right? You're going to allow a great deal of fluid inside this space, but it has to be under a certain amount of pressure so it can actually exit the space and collect blood back into the capillary bed, or collect fluid back into the capillary bed to now exit through that efferent arteriole. Okay?
So it's regulated by the volume of blood that comes through and not by opening and closing.
It's going to be a mixture of three things, but the myogenic response is one of the first ones that actually causes the whole system, because the myogenic response is determining the amount of stretching inside that space.
Okay.
Okay, cool. Now this is what an actual renal corpuscle looks like. If we were to take your kidney, take a slice of it, and actually take a look at that capsule, you don't see this cartoon here. What you see looks like a bunch of cuboidal cells surrounded by some blood vessels, right? But that's actually what's happening. And in this little tiny space here, that's where the fluid is moving into, such that it will actually exit out now through that proximal convoluted tubule. Now, a couple of things I want to make sure you pay attention to before we leave here. Mesangial cells, podocytes, afferent arteriole, and the innervations of the sympathetic nervous system, as well as that macula densa. Okay, so when you're reading and you come across these words again, make sure you're saying to yourself, oh, he stressed that we actually need to know that stuff. Okay, cool.
Sorry, repeat that again. The podocytes, the afferent arteriole, the innervation system, and the macula densa.
Correct. Don't forget the mesangial cells, though, okay?
Yeah.
Okay, cool. Great. Okay. So how does this all work? I just skipped over all the words. Do you guys really want me reading slides to you?
Not really.
I didn't think so. Because you can read yourself, number one, but number two, unless you're going to see if this is the blood traveling in, we're going to filter the blood in from the plasma into this space. It's going to travel through the proximal convoluted tubule, blah, 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 yada, yada, yada. What I want you to pay attention to is that inside your textbook, they're essentially saying the same thing that I did with that more important slide at the beginning. Is depending upon where we are, we're going to move unique combinations of ions depending upon the epithelial cell. Okay, let's just make sure. Okay, filtration. Daily volume is, you're actually moving between 150 and 180 liters of plasma via tubular reabsorption each day. Okay, so this means you've pushed it into the glomerular capsule, it's made it into the proximal convoluted tubule, and then somewhere in that tubular structure, it's going to be reabsorbed back into the venous blood return. That's what that means. Now, how many liters of blood do you have in your body? Quick.
5.
Fantastic. Take a look at how many you're actually reabsorbing in the course of a day. It's a lot of heartbeats. Okay? With me? Cool. Now, the way it, of course, works is we're pushing everything across endothelial cells. Okay? And because those podocytes are actually encircling the capillaries, it creates a filtration system for small solutes to actually move in the space, but it prevents plasma proteins, blood cells, and platelets. So it has to be that small that it doesn't let platelets out. So think about that. Think about the size of a platelet versus the size of a red blood cell. And it's going to actually cross 3 layers, the glomerular and epithelial cells, the fenestrations, and then finally getting into the filtration slits of the podocytes themselves.
All right.
That
Can you
looks like
say that one more time? So it,
Sure.
the 3 layers are the podocytes, the
Yep.
endothelial cells,
Yep. And one more. So endothelial cells, the basal lamina of the end endothelium, and then the filtration slits of the podocytes themselves.
Okay.
Okay? And that's what you actually see here. So here's a podocyte. It has these projections. They're called pedicels, whatever. But what I want you to see is the space. What Mother Nature is doing is she's reducing the total amount of space, and she's creating, look at the type of capillary bed she's building for you guys. Anybody remember what? These are fenestrations, right? So this allows a lot of material to pass through, but not so much. The size restriction stops at around the size of a platelet. And that means there's actually pressure to move materials into that space. Okay? Nudge, nudge, hint, hint, wink, wink. All glomerular capillary beds are fenestrated capillary beds. Okay, cool. You're like, okay, now I know he told me that for a reason, and that's because he already said we're going to generate pressure. Pressure seems to run everything inside AMP2, doesn't it? I'll just take that as a yes. Okay. Okay. Where the net filtration pressure, is the glomerular blood hydrostatic pressure in the blood of the capillary beds minus the capsular hydrostatic pressure. So blood moves into the space, the capsule pushes back. Okay. And once you've pushed all those materials through that fenestrated capillary bed, you now have that opposing blood colloid osmotic pressure you were hoping you were never going to see again. Remember back in chapter 21, we had to get fluid back into your circulatory system? We had blood colloid osmotic pressure there. It works exactly the same way now. Yes.
All right.
It's like you're talking too damn fast.
So remember when you gave us a drawing of the capillary bed and you showed the hydrostatic pressure, and then you showed.
It's exactly what I wanted you to think about. Zach, do you want me to draw you a compare and contrast? I can do that for you. Okay, let's go like this. So here's the capillary bed you're used to seeing, right? Remember, blood travels into this space at roughly 36 millimeters. I hate when that happens. Sorry, I'll be right with you. Blood travel. I'm sorry?
So I think I need a mask.
Yeah, exactly. Oh, we're all going to die. No, no. Trying to think of what movie that comes from, right? What happened? I actually can't think of it, right? So remember, blood comes in here at roughly 36 millimeters of pressure, okay? And that the blood hydrostatic pressure is what's pushing out into the space. But because fluid's traveling with it, it means now we've concentrated everything inside this vessel, and that causes the blood colloid osmotic pressure to pull things back into the capillary beds.
Yes.
Let's come over here. Let's draw this. Don't hit that button, Jonathan. It will all go away again. So let's draw this for you. So here's a capsule, proximal convoluted tubule. Okay, put that there like that. Put that there like that. Endothelial cells, yay. And in red, here comes our blood vessel. Afferent arterial, glomerular capillary structures with everything that's required on it. And here is our afferent blood flow, and here's our efferent blood flow. Okay, so what happens here is the glomerular blood hydrostatic pressure travels inside this space, and that's what pushes out. Okay? The capsular hydrostatic pressure is literally, because this actually travels all the way around it, I should surround it all the way up to the blood vessel there, right? What happens is the fluid that moves in here now literally pushes back onto the capillary bed. Okay, but what it does, of course, is it allows for us to generate force that's going to allow the filtrate to now move into the proximal convoluted tubule.
Okay.
Simultaneously, though, because that's happening, remember, we've removed all of the volume inside this space. So now all of your fibrinogens, your albumins, all of those are now needing more solvent. And that's what causes the blood colloid osmotic pressure to pull fluid back into the circulatory system. And that's what actually travels out through the efferent portion. So, okay, so literally, Desiree, what you're doing is we're just looking at this side of a capillary bed inside of a glomerular capsule.
Okay. Much better.
Good, I'm glad. No, because this is the basis of generating pressure to now push everything through that tubule. It has to be an appropriate pressure for everything we've discussed before, because otherwise the epithelial cells we're about to look at won't have the opportunity to move the materials back into be reabsorbed. Okay. Voila. So here's the cartoon. Blood comes in, 55 millimeters of pressure here. So it's not 36 millimeters of pressure. And what's going to happen is we're going to move into this space, and the capsular hydrostatic pressure pushing back is 15 millimeters. The blood colloid osmotic pressure is now 30 millimeters. And notice what this gives us. This gives us a positive value. It's that positive value that actually pushes the fluid now through the proximal convoluted tubule to begin the true filtration process that's going to happen inside the tubules. Make sense? And this is where I was saying earlier I should have had the capsule come right up alongside the vasculature so you could see that.
Yeah, but when you look at where it says efferent arteriole, you have the opposite direction. So what's making that happen?
Right, so there's still blood pressure inside of here. It's not like we dump all of the fluid inside of this space. Some fluid remains. Not only that, remember our blood colloid osmotic pressure, that's pulling the fluid back into the capillary structures, and that's the volume that's actually exiting here. Okay?
Okay.
Cool. Woo-hoo. Boink. Okay. So, homeostasis requires we maintain a roughly constant glomerular filtration rate. Too high, substances pass too quickly and they're not reabsorbed. Too low, nearly all are reabsorbed and nothing is excreted. That's just as dangerous as actually not retaining anything inside the secreted materials at all. So how do we actually regulate now the glomerular filtration rate? Just like we did inside chapter 21 or we did inside chapter 20, we have to figure out what's going to maintain the adequate amount of pressure inside this space for filtration to occur. Wah, 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 wah. I'm talking too fast. I'm ahead of schedule here. Sorry. I'll slow down now. Now that I know I'm going to hit my time hack, I'm just going to sit back and relax. We will discuss renal autoregulation. Okay? And what you're looking at here is the kidneys actually maintain themselves. You set them up, put a little, fluid moving into them, and they're just hunky-dory. You don't really have to do anything moment to moment inside of your life. A way to think about it is this. Say your phrenic nerve is responsible for causing inhalation, but normal respirations don't require any musculature for exhalation. Your kidneys pretty much function the same way. And that's because as your heart is beating, remember, it's causing blood pressure. That blood pressure actually causes a myogenic mechanism. That's going to stretch the arterioles, okay? And because what it's doing is it's stretching the smooth muscle, that will temporarily reduce the glomerular filtration rate, okay? you have, as a result of this, or in conjunction with this, is what probably the better way to say it, is you actually have a tuboglomerular mechanism. And remember I said you wanted to pay attention to that macula densa? Okay, remember, it's literally where the vasculature is adjacent to the capsule. The macula densa provides feedback to the glomerulus. Remember, Desiree, you're asking what regulates vasodilation? That tuboglomerular mechanism inhibits the release of nitric oxide. Okay, so somebody think that one through with me out loud. Nitric oxide causes what?
Vasodilation.
Fantastic. Now, if you inhibit the vasodilation, what actually happens to the tonicity or the rigor of the actual capillary bed or the smooth muscle?
It would constrict.
Even if it doesn't constrict, at least it doesn't dilate.
Right.
Okay?
Or it'll go back to its normal.
Exactly, right? So what happens, of course, is this is going to cause the afferent arterioles to constrict as a result of that. And that's not even including any sympathetic input. Okay? So moment to moment, stretching and the inhibition of nitric oxide regulate the auto-regulation piece of filtration.
You have to say that again.
I will,
Sorry.
sure, no problem. Okay, so every time your heart beats, right, you're going to cause a change in pressure. That pressure is going to stretch the smooth muscles that are surrounding surrounding the afferent arterioles. That's going to reduce glomerular filtration rate. What happens is you now have an antagonistic mechanism that's using the macula densa to say, hey, the pressure's too low or the pressure's too high. What it does is it inhibits the release of nitric oxide. That nitric oxide then causes the afferent arterioles to constrict because it's not allowing vasodilation to occur. That's a pretty nifty mechanism because you think about you're doing that for 180 liters of blood a day without fail. At least till you get old like me. Then all of a sudden you're like, what the happened to my kidneys, man? Okay, cool. So that's just the auto-regulation piece. Okay, that's just step one. Step two, of course, well, let's make sure everybody understands the feedback system here that I plucked out of your textbook for you. Notice the glomerular filtration rate. What it's doing is it's measuring at the juxtaglomerular apparatus. Oh, where have we heard that before?
I'm sorry, repeat the question again.
Where have we heard juxtaglomerular apparatus before?
Is it in the respiratory?
Unfortunately, no, young lady. Remember, we used it for our RAA pathway?
Oh, yeah.
Yeah, oh my God, you were thinking you were going to get through the semester without ever seeing that again. Sorry, no, but we'll come back to that later. Because the juxtaglomerular apparatus in the macula densa detect some change, increased delivery of sodium, chloride, and water, what happens is now you decrease secretion of that nitric oxide, and that's what actually causes the vasoconstriction. Okay? So we're going to tie these two systems together. You just don't know how they're about to be tied up. Now, the neural regulation, this is pretty straightforward, right? The kidneys have blood vessels that are supplied by the sympathetic autonomic nervous system fibers, and norepinephrine causes vasoconstriction. Okay? You're like, okay, I got that. Woohoo. And normal or moderate stimulation will cause both afferent and efferent arterioles to constrict at roughly the same degree. You're like, how does Mother Nature do this? Well, she releases the neurotransmitter and they vasoconstrict simultaneously. However, if imagine something scares the tar out of you, right? that pulse of norepinephrine will cause the afferent arteriole to constrict more, and therefore you essentially drop glomerular filtration rate to 0 because no blood is then moving into that glomerular capsule. Okay? And what stops sympathetic stimulation? Nothing. You just return back to normal. And this is why after you've survived scaring yourself with your own shadow, okay, your kidneys go back to auto-regulation. They're like, man, you're such a ninny sometimes, Jonathan. Your shadow shouldn't scare you like that. Your kidneys are going, nah, 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 right? Okay, no? Anyway. Now, we can also regulate the the glomerular filtration rate at 1 and angiotensin 2 concentration. Remember, angiotensin 2 is a potent vasoconstrictor, okay? And what it will do is it will close both afferent and efferent arterioles. And you can see where we're going with the RA pathway here, right? We skipped over the renin, the angiotensinogen to angiotensin 1, and angiotensin 1 gets to angiotensin 2. But look, there's our angiotensin 2. Okay, so even though we may not have directly said we activated the RAA pathway, somehow we made angiotensin 2, and now we can cause vasoconstriction at both afferent and efferent arterioles. Now think about this for a second. We've just stopped filtration inside your kidneys for some period of time, which means all of the blood that would normally be passing through your kidneys is now being kept inside your systemic circulatory system. And this is the major mechanism for retaining blood pressure inside your body when the RAA pathway has been activated. Okay? Now, in those moments when you have way too much fluid inside your body, and remember your right atria is stretched, we release atrial natriuretic peptide, ANP. Ding. ANP makes you go to the bathroom. Ding. Right? No. Whatever. Anyway, it's going to increase the filtration rate, and that filtration rate is going to now increase the capillary surface area to allow for more filtration to occur. Okay. Cool. So now the reabsorption piece, if filtration has occurred and you understand the mechanisms now, whoops, sorry, I think somebody's been sitting there for a while. Sorry, Gia. Thanks. Okay. Okay. If reabsorption is going to occur or secretion is going to occur, both of these actually have specific reasons for occurring, right? Most of the reabsorption for, say, water occurs in the proximal convoluted tubule. Roughly 99% of the water that's filtered into that capsular space is ultimately reabsorbed inside the proximal convoluted tubule. Whereas what we do with secretion moment to moment is help control blood pH. And what's the major ion we regulate when we're controlling blood pH?
Hydrogen.
So hydrogen protons are definitely one molecule that we control that is an ion. Give me another molecule or compound that we need to control that isn't an ion.
That isn't an ion?
Correct.
CO2?
Carbon dioxide, awesome, right? Because carbon dioxide with water, which we have a ton of, right? In the presence of carbonic anhydrase is going to make bicarbonate. And that bicarbonate will actually help us regulate blood pH. Look at that. You guys really are going to save people's lives. I've got hope. Because we tied this all together inside chapter 27. Woohoo! You're like, 27? We haven't finished 26 yet. Okay, well, we're going to finish off this evening. It's like 6:15. Like I said, I'm about 10 minutes early. Okay, you're like, what? We just started. No. You need to go back and read these mechanisms, right? How do we actually reabsorb materials? Did anybody understand this when they were reading it inside the textbook? Did anybody read the textbook yet? Look at that. Oh, you did. You're such a liar. You're like, no, he's not going to see me say no, but then I'm going to say yes, right?
I think this is where I got incredibly confused because it was like term after term, and I was like,
right.
So lost.
Okay, cool. So I'm going to try and clear some of this up, and then I'm going to let you guys go. So we pick up here again. So when I walk through the important ones, the hydrogen, the carbon dioxide, the sodium, the chloride tomorrow, you'll understand exactly what we're doing. But we have major reabsorption routes where primarily the mechanism is paracellular reabsorption. And that means between adjacent cells. So if here's a cell and here's a cell, it means some materials can pass through them. And we use tight junctions, don't completely seal, and therefore it is a passive mechanism. And what actually creates the pressure, I should say that creates the movement of the fluid, is the pressure. So the closer you are to the proximal convoluted tubule, the more likely you are to use water pressure, or the pressure inside the tubule, to push materials through. You also will collect large amounts of material, namely, let's say, glucose, okay? Using individual cell transporters, glucose transporters, that are not insulin dependent, okay? So you can reabsorb all of that glucose and put it right back into the body and not lose the regulation there, okay? That's really important, because what it means is we tied together the area where you have the highest amount of pressure to move probably the most important materials back into the circulatory system by reabsorption. So how do we actually transport things then? Notice the ion that we list here first. The major reabsorption of the ion sodium is how we regulate water volume inside of the body, right? So by moving sodium, it means we use primary active transport, secondary active transport, and these are regulated by how fast they can work. So let me give you an example. What are you having for dinner tonight? Anybody? The kids are on their own. I don't care what they eat. I'm gonna read my textbook.
Cereal.
Cereal, fantastic. Okay, and now, and when you look at the side of your box on your cereal, you're going to notice that most of those are actually salts that are actually inside of your cereal, right? Do you ever like eat a bowl of cereal for dinner and wake up with crusty eyes? No, I've never thought of it before. Do an experiment tonight. You know, make a bowl of cereal, eat it. When you wake up in the morning, find out if you actually have crusty eyes. And what's happened there is you've ingested so much salt. Okay, you've ingested so much salt that what ends up happening is inside that. Oh, look at
sorry.
that. That's okay. That's wonderful. Okay. Okay? So what's actually happening is you're ingesting so much salt that your kidneys can't process all of it, so the tear ducts do. And that's why. Imagine, if you will, another example, not just cereal. Imagine, if you will, you went out and you had fish and chips with, the chips are loaded with salt on top of them. The same thing would happen, but I threw in the fat there because your body needs to process the fat as well. Primary active transport is that sodium-potassium pumps on the basolateral membrane only will move sodium. That's only where we use primary active transport. Secondary active transport is when we actually use symporters and antiporters. You haven't thought about symporters and antiporters since AMP1. So tomorrow when we start, we come back with explaining how do symporters, how do antiporters work, okay?
Well, which of the ions are using the symporters and antiporters?
Yeah, no easy answer for you there, kiddo. That's why you have to wait for tomorrow. Okay, but if sodium is one of the ions that actually has to be moved, it's going to use both, and you'll see how, okay? And it's going to do this because depending upon where we are in the nephron, we'll use a symporter, we'll use an antiporter, or we'll use primary active sodium-potassium pumps in specific locations, okay? Now, again, this may be an anatomy and physiology class, but this is not a renal physiology class. We're not asking you to know exactly which 3,000 cells in the ascending limb of the loop of Henle are responsible for what concentration of sodium gets pulled out. But we do hope that you understand this. We do hope that you understand by the time we finish that moment to moment, there is a transport maximum that can actually happen. And that's determined by the flow rate. There's only so much material you can move across the surface of a cell. There's only so much material you can pull into a cell and then transport by transcellular movement.
And that's why sometimes if they find you have sugar in your urine, that's a sign that something's going on.
Well, that means a lot of different things. I mean, it means that maybe up here in the proximal convoluted tubules, you either have hypertension, the pressure's too high, or the transporters are broken and there's no way to actually get the glucose in, or there's some sort of traumatic inflammation. Okay, some UTI has traveled up far enough into your kidneys where the inflammation now stops all of the normal filtration mechanisms. Okay, cool. So I just want to show one more picture and then we'll be finished for the evening. And here it is, right? So yeah, so yes, you get to this part of your textbook and you're like, okay, I'm ready for almost anything. And you're just like, huh, what's going on here? And this is one of the easier figures, right? This is just simply saying that in some instances, sodium can just simply pass through tight junctions and go return back into your vasculature. And notice it's in red, suggesting it's a peritubular capillary bed, and it's passing through that interstitial fluid. Yay. On the basolateral membranes, these are primary active structures, and these are the sodium-potassium molecules. But remember, it's active transport, so it actually takes ATP to now move sodium against its concentration gradient. Not that it's really moving against its concentration gradient. Okay, but the point is this: one is diffusion, one is active transport, the other, oh, take a look at our cell here. I mean, our pump. What is our pump name again?
What do you mean? What do you mean? It's an active process? It requires...
Yes. Well, no, so the sodium potassium ATPase does require an active process, but what hormone regulates those? Thyroid hormone. Okay, so if you have a decrease in T3 and T4 inside your bloodstream, you have fewer sodium potassium ATPases inside the lumen of all of your possible reabsorption routes, which means then you lose the ability to regulate sodium inside your body. Okay, so if your nervous system is a little bit sluggish, now you know why. You have insufficient sodium for the correct generation of action potentials and or resting membrane potentials inside cells. But you see more of this along the time when you're about to die, not so much that. But this is a really good place for us to pause, right? Because what I want you to do is go back, look at the construction of those two different nephrons, make sure you understand the distinction between the cortical juxtamedullary nephrons, and pay attention to the flow of blood into the capsules, coming from the renal artery, exiting out through the renal vein. So when tomorrow we pick up, we'll speak about what it means to use nitric oxide and norepinephrine to change the actual flow rates before we finish off chapter 26. And possibly tomorrow, we could finish 27. You're like, what? I haven't started 27 yet. But you've started and finished 27 already.
Then we have no class on Friday.
No, we have review on Friday. Unless, of course, you don't want to meet on Friday. I could use a gin and tonic at 5:30 in the afternoon instead of talking to you guys. I really could. Maybe I'll make myself one while I'll be like, and now my kidneys are failing me. Okay? But go read, try to get to that figure that I started class with, right? Get to this one. Let's see, escape from here. Escape, Jonathan. Get to this figure so that you can actually see what I'm talking about now. Notice every single step of the way through our kidneys, we regulate sodium, okay? But there's a specific different mechanism that's using that. Now, like I said, it's not a renal physiology class. I'm not asking you to memorize these differences. What I'll be teaching you tomorrow is why Mother Nature is moving each of these when she's moving, okay?
Can you put the videos up if you possible?
They already should be. There should be a link to YouTube. Yeah.
Tonight as well, if
of course.
you can.
I can do
Okay.
that as soon as we get offline.
Okay, thank you.
No problem. Okay, folks, go eat some dinner. Make sure it's not just cereal. Okay, and if it is, do your experiment and see if your eyes wake up crunchy in the morning. We will wait for a result. Okay.
I'll see how it goes.
Yeah, she'll like, no crunchy. We're good.
Thank you. Eat something else.
Exactly.
Thank you.
Have a wonderful night, guys. Take care now.
All right.
Bye.
Professor, are you still able to meet?
Yep. In fact, I'm gonna wait till everybody leaves, and then you and I will chat. Okay.
Okay.
Shut off the recording.